Here we are at part 2 of this chapter, and we will be exploring two techniques which are used, they're used quite commonly in a lot of different arenas in biology, but we will be talking about it here in the context of criminology. And their purpose is, in particular this context, for what's known as DNA profiling. DNA profiling is being able to look at various individuals' DNA and use the unique properties of our DNA to help identify an individual of some kind based upon their DNA for whatever reason. And then we'll talk about some of those reasons here in a minute. But the first technique that we're going to talk about is the polymerase chain reaction. And this is a very commonly used technique, and it's used to amplify DNA. And what we mean by amplify DNA is you can take literally one molecule of DNA and you can, in a very short amount of time, make a very large amount of that DNA. So it's basically just making many, many copies of DNA, and this is exactly what is done on many COVID tests. We've heard PCR before. How many PCR tests have there been conducted for COVID? And basically what they'll do is, the way that it works is that the virus that causes COVID-19 is SARS-CoV-2, and that has a nucleic acid sequence to it. And so if that sequence, that very, very small sequence, is found anywhere in your saliva or your nasal mucosa or anywhere in your body, it will be present in a sample that you take of your body, whether it be a spit sample or nasal swab. And even if just one copy of that specific sequence that is carried by the virus is found in your body, PCR will make many, 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 many millions of copies of it. And then it makes it much easier to detect if, in fact, that sequence of RNA is in your body or not. And if that sequence of RNA is in your body, then it's likely the case that it's in there because you at some point had a virus or currently have a virus that's replicating inside of your body. Okay, so how is this done? It sounds a little like witchcraft, but I assure you it's not. It relies on a couple of things. There must be some sort of DNA template, or it could be an RNA template that's eventually converted, but there has to be nucleic acid, and specifically the region of nucleic acid that is of interest. So in the context of a COVID-19 test, the region of nucleic acid that would be present would be a portion of the viral genome that's packaged up inside the virus. Okay, then all that's required is you have to add nucleotides, which are these. Nucleotides are all the building blocks that will link together to form DNA. You have to put primers on there because we didn't really talk a whole lot about this, but DNA replication always begins when a primer, and then from there, the polymerase can bind and start replicating. Okay, so these are some of the three ingredients. You need the primers, you need the polymerase, and you need the nucleotides. The specific polymerase here is called a Taq polymerase because it comes from a specific type of prokaryote where the ability for this to turn on and off is temperature dependent, and it can also withstand high temperatures. Okay, so here's how it's done. The first cycle looks something like this. Basically, the first step is that the nucleic acid would be denatured. So in other words, this has to be split into two. So these bonds that keep the two strands, if it is double-stranded, the bonds that keep the two strands together have to be broken so that this can split apart. And then that will allow the primers to get in here. The part is a temperature. So the temperature will go way up. I forget what the exact number is, but it's like 95 degrees or something like that, Celsius. Okay, then the temperature is brought down to 60 degrees or so, and because the primers are smaller than the big chunks of DNA, they're able to quick get in here and bind faster than the original strands can then come back together and rejoin with one another. And as soon as the primers get on there, then it's going to keep these, it should keep these two pieces of nucleic acid from coming back together again. So you open up the double strand, you quick put some primers on, and then that should keep everything open. The annealing is done by dropping the temperature down. So first the temperature of the whole mixture here is brought up to 95, and then it's brought down, I think it's to like 60. The actual number is not important, but I think it's something like that. Okay, and then what's going to happen is that the polymerase will bind to any region where there's a primer. A polymerase always has to bind to a place where there's an RNA primer. And then it just goes through and it literally just replicates the DNA just in the same way that it normally would in our cells when they're undergoing the S phase. We're just making that happen here for this little small chunk. Okay, so now we've started with one fragment of DNA, and then we've ended up with two. And then we can just repeat the whole cycle again. So the second cycle, again, you'll open these up, the primers will bind, and the polymerase will then replicate it. So now we ended up with 4 because we got 2 from each of the ones that were there at the end of the first cycle. And then after this, the third cycle, each of those makes 2 copies. And so we can just keep going many cycles. Oftentimes, in the past, when I've done this, it's usually around somewhere between 20 and 30 cycles. So if you were ever interested to know how many copies of DNA that would be, there'd literally just be 2 to what either 20 or whatever number of cycles were completed. And that ends up being a huge amount of copies. So and this happens quite rapidly in just a very quick amount of time. One copy can be made into many, and like less than 20 minutes. In fact, I actually just had to get a COVID test for my job. They now require that I get one in order to stay employed there. So it was pretty cool. They had like this little machine, you put like a big white chip into it, and then there's the nasal swab, and you literally just stuck the nasal swab into the chip, the microchip thing, and then within like 20 minutes the result was given. So I think that what that machine was, that little chip that I put the swab in, it was a thermocycler, or it was the machine that does this reaction, and it was heating it up, and it's basically the nasal swab probably went into a solution that had all of these primers and nucleotides and polymerase there, and it literally just heated up and cooled it down, heated up, cooled it down, heated up, cooled down a whole bunch of times, so that within about 20 minutes there was a whole bunch of these that would have been made had the original nucleic acid sequence from the virus been in my nasal cavity, in my schnot. Okay, and then yeah, so it's just like a little in-house little machine that does all of this, I think, is how it must work. And then it's much more easy to determine if there's the viral genome present in my body based upon how many copies of this would have been made. It can be more readily detected. Okay, another technique that's very commonly used and actually will also employ the polymerase chain reaction is gel electrophoresis. Gel electrophoresis is what allows us to create a DNA fingerprint, or in other words, because all of our DNA is unique, if we chop it up at certain spots, it will create different size fragments, and then we can actually pull those fragments through a porous gel and separate out all of the different fragments based upon their size. And so because we all have different size fragments that are made, there'll be different bands of DNA that show up on a particular gel, and then you can compare the different types of DNA. So how this is done, it utilizes what are known as restriction sites and also restriction enzymes. So a restriction enzyme is a protein that cuts DNA, and it cuts DNA always at a very specific spot, which is called a restriction site. And there are lots of different restriction enzymes, and each has a sequence of bases that it will cut at. So you can kind of alter which restriction enzymes you want to use based upon which restriction sites you want to cut. But because our DNA is all different, the place where our restriction sites end up for any particular restriction enzyme varies. For example, it might be in my DNA there's a restriction site right here for a particular restriction enzyme. And then if that was the case, I mean, if it truly cut it right here, that would result in this little piece. So I'd have a little piece of DNA here, and then I'd have this really, really, really long piece like this. So this, if I tried to separate this out on a porous, by dragging it through a porous gel, then I would get two different bands, one that's composed of the little fragments and one that's composed of the big fragments. The big fragments would take longer to get pulled through the gel, so they would be closer to the origin, and the shorter fragments could sneak through that gel, that porous gel, a lot faster, and then they would migrate further in the same amount of time. Otherwise, like if we, like on your DNA, if your restriction site came here, then if we cut that, then it would lead to two fragments that are more similar in size, and so there'd be two different bands. And ultimately, there's actually a lot of different restriction sites in different places, so it makes many different sized fragments of DNA that can get pulled through a gel. The way that a gel is made for electrophoresis is we use a catheter, which is illustrated here. So this is just like a piece of plastic, and then you'll basically tape off the two ends here and here, and we're actually labbed, so you'll get to take a crack at it. And then they put a comb in there, and then they pour what's known as agarose into the casting tray. Agarose is like it's made out of agar, which is like a molecule that's derived from seaweed, and it's in hot temperatures it's all dissolved and it's a liquid, but then as it cools it solidifies, and the molecule of the molecular structures in agar or agarose are such that when it cools it creates pores, so it allows stuff to actually, molecules to actually get moved through. And then again, the premise is that it's the smaller molecules that will move faster, and then the larger molecules, they move much more slowly because it's harder for them to wiggle through all of those microscopic pores. Okay, now what happens then is that once the gel is, once the gel is made, so once this agar solidifies, they take the comb out and it will leave little wells in the gel. And then it's into those wells that DNA will be dispensed. We use a tool called a micropipette, which is basically an instrument that allows us to administer a very, very, very small amount of volume. Usually in this case it's about 20 microliters, which is a very small amount of volume. Okay, so what we're seeing here is they're actually loading DNA into those wells. And the DNA is purple because it's dyed with a loading dye, and that allows us to basically see which of these wells has DNA in it. The loading dye also makes the DNA heavy, so it sinks down to the bottom of those wells that are made. Once all the DNA is loaded up, and keep in mind at this point the DNA that is added is DNA that likely has undergone the polymerase chain reaction, so there's a lot of whatever particular DNA we're interested in evaluating, and it's also been digested, so it's been cut with restriction enzymes already. Okay, so this in here is amplified DNA that has been cut with restriction enzymes, so already the fragments of DNA that are going to be present are present. Okay, and then we load this gel into something that looks similar to this. This is called a electrophoresis chamber. Electrophoresis, because what it does, it is actually there's two electrodes. There's the positive and the negative electrode. I can't ever remember which one is which. I think this is negative and this is positive. And what happens is that electricity flows across this way through the fluid. There's actually fluid in here into which we'll put the gel, and you can actually see there's DNA dispensed in these wells. And as the electrical current passes through the gel, it will pull the DNA across. This happens because DNA is negatively charged because of the fact it has that sugar phosphate background. Well, phosphate has negative charges on it, so all in all there's a slight negative charge on molecules of DNA. Well, when we apply a current across here, the negatively charged DNA is attracted to the positive field of the electrophoresis chamber. And so it drags, for lack of a better word, the DNA fragments through the gel, and then those that are the smallest will migrate faster, and those that are slowest or biggest will migrate slower. Basically separates them all out based upon their size. So it looks something like this. The bigger they are in terms of base pairs, the closer they will be, and then the smaller that they are, the greater the distance they will migrate. So in other words, you might start off with putting some samples into the wells, and then usually also when this is completed, we put a marker in one of the wells. The marker is a sample of DNA that's been cut a certain way so that we actually know the size of the DNA fragments that will congregate to form all the different bands after we run the gel. So let's say we run the gel, we turn the electrophoresis chamber on, put an electrical current through it, it starts dragging all the DNA fragments through. Then you should start to see, once it's all done, a separation of the DNA into the various sizes that it is. And then the marker will tell us for any DNA sample that we run. Let's say we run it and there's a sample of DNA fragments here, there's a sample of DNA fragments here, here, and here. Sample after we cut it with the restriction enzyme and ran it through the gel, it made 4 groups of DNA that all varied in the size of the fragments in those groups. We can then figure out the relative size of the fragments in each of these groups by comparing it to the ladder, is what it's sometimes known as, or the marker. So in this case, if I was wondering, okay, I see a band of DNA here on the gel, this means that in this region of the gel, a whole bunch of chunks of DNA are here, and I'm wondering how big the size of those DNA fragments is. I can just come over across, and we see that it lines up with this particular band. So anything that's in this region likely would be something that is of a size of approximately 600 base pairs in length. Or similarly, if I wanted to know how big the DNA fragments are in this chunk, I can come across the ladder and I can see it's somewhere in between 300 and 200 base pairs. So maybe it's about 250 base pairs is the size of each of the DNA chunks in this group. Okay, so pretty cool, and this again allows us to do all sorts of stuff. So for example, here's a picture of a gel that has been run. Here we see the marker. Here we see all the bands, and they've labeled them based upon the size in kilobases. So like 4,300 kilobases or 3,500 kilobases. So they've labeled the size of each of the DNA chunks that are in these bands. And then they've also put some sort of fluorescent dye on them of some kind and shining some sort of light on them so they glow. I don't know exactly what they put on them or what kind of light they use to make it glow like this. But here you can see, like, let's say this is DNA, just for the sake of example. Here, the DNA that was in this well was DNA that was found and isolated from a murder weapon at a crime scene or something. Okay, and then there was 4 suspects, or there is 5 suspects. So we put the suspects' DNA in here. We treated it with in the PCR reaction to amplify it, and then we put it in these wells. We cut it with the same restriction enzymes that we cut the DNA that was found at the crime scene that was also amplified. And then we run it all out. You can see that this suspect right here yielded DNA bands that fall on the same location as the DNA that was found in the crime scene. So this would suggest that this individual here has the same DNA as the DNA that was isolated at the crime scene. Now, it doesn't necessarily mean the individual is guilty. It just means that the DNA that was found at the crime scene or on the murder weapon or what have you is the same DNA as the individual. Whether it was them that actually did the crime, it's harder to determine, but something like gel electrophoresis can tell us at least if this particular individual was at the scene of the crime or if their DNA was truly found on a weapon of some kind or something like that. There are a lot of different applications for this that go above and beyond just solving crimes. These can be used for paternity and maternity tests, so you could run the DNA of mom, dad, and baby and see if baby's DNA more closely matches mom's DNA or dad's DNA. If there's a match there, that would help to confirm if a child really did in fact belong to a parent or not, or if there is another potential thing that happened. Okay, as we said earlier, it can help to ID crime and forensics. For organ donation, to see which donors might have a genome that's most similar to somebody that needs to be receiving an organ, they could use this test. They could use this to diagnose inherited disorders. So you could take the blood of somebody that has, for example, hemophilia, and amplify their DNA from their genome and run it, and then if somebody else had that same mutation, it would likely be the case that there was a matching band pattern on the gel, depending on how it was cut and treated and run. Okay, test for pathogens in foods. So you can take food and grab the DNA and amplify it and see if it matches some sort of pathogen that has a known banding pattern for some particular protocol that was used to digest the DNA and sort it out. Okay, you can determine if something is genetically modified or not. So, and then also comparative biology, basically seeing how similar the genomes are between different animals that maybe live now or didn't live now or are extinct or whatnot. It's powerful because one only needs a very, very small sample of DNA to do this test, because PCR allows you to take one simple small molecule of DNA and make gazillions of copies of it, and then once it's amplified, it's really easy to cut it and run it and separate it out and see how it looks and compares to other things. In either case, it's a pretty simple test to do. We're going to do it in lab, and this is something I did a lot when I worked at the U of M, and a lot of different research groups use this technique for a variety of reasons. So very interesting little piece there of biotechnology that we can use, and interesting to talk about it here in the context of criminology. This concludes our discussion of the use of biology in criminology.